Okay, welcome to The Issue. I'm Jason West, and I'm joined here by my new co-host, Sanjay D'Souza. We're at the ASGCT 2026 annual meeting in Boston. And today, this is a special episode because we're going to turn the microphones on ourselves. As new members of the Issue team, we wanted to take this opportunity to introduce ourselves to you, our listeners.
Hi, I'm Sanjay D'Souza, your co-host. We're super excited and humbled to be stepping into this role as co-hosts. Emily Walsh-Martin leaves behind some very big shoes to fill, and we're grateful for the foundation she built for this podcast and the community.
So today is really about introducing ourselves to the listeners, who we are, where we came from, and where we hope to take the issue moving forward, and to solicit help from you, our listeners. What would you like to see covered in future episodes? Who would you like to hear? And how can we help advance the cell and gene therapy field?
But first, applications are now open for ASGCT's early career funding opportunities. ASGCT members are eligible to apply for two $100,000 grant pathways, the Career Development Awards and the Expanding Pathways Fellowship. Applications are due August 1st, 2026. Please visit asgct.org/awards to learn more.
Cool. So first up, I'm going to ask Jason a few questions. Jason, is an honor to co-host with you. And I'd like to ask you a few questions so that everyone get to know Jason a little bit better. So the first question is, what first drew you into science and biotech?
Yes. Thank you, Sanjay, for the question. Like many, I think my entry in the field in retrospect sort of looks a little bit more random than rather like a mission. Like I've been lucky in that my family doesn't have any rare genetic diseases in it. I grew up in West Virginia, went to undergraduate at West Virginia University, and at the time I was looking for ways to live in a city, to sort of get out of the country. I love outdoors and nature, but was looking for more of an urban experience, and was lucky to stumble across a Howard Hughes Medical Institute summer program at the University of Pittsburgh. And so being driven by an interest in science, I applied, was delighted to be accepted, and got to work in my first lab experience in a bacteriophage lab with a professor who's now deceased named Roger Hendricks, who just did an amazing job sparking curiosity in his trainees and providing a foundation of discovery and learning. And I think that really put the hooks into me for a career in science, particularly in early discovery, where I've worked most of my career, either in the academic side or now in industry. From Pitt, I went on to find a lab at WVU that was studying how human bone forms and absorbs itself. Went on to find other summer opportunities in urban environments, including a summer undergraduate research program at NYU, where I got to live in New York. Really enjoy city life while balancing it with the same framework for discovery in a lab, studying transcription control with Michael Garabedian at the medical school there. From there, I went to graduate school, which brought me here to Boston, where I still live. I did my PhD at Harvard in stem cell biology, first using embryonic stem cells as a tool for developmental questions. I then went to MGH, studied with Bob Kingston on how genes are turned on and off during using stem cells as a platform for that discovery. And then, like many during their postdoctoral career, I had to start actually thinking earnestly about how am I going to make a career out of this amazing training I'd been provided. I had many friends working in industry, and that led to my transition to my first industry job at Amgen, where I brought the stem cell platform to, again, model disease and normal biology to try to nominate drugs for targets for druggability. So did early target nomination, target discovery, and then worked on some drug development programs there. From there, I was using gene editing as a tool, but never as a therapeutic modality, and was very lucky to land into a group at Biogen using zinc finger nuclease technology as a tool. And so for the first time, I started to work on therapeutics in earnest in the cell and gene therapy space. Got to attend my first ASGCT meeting back in Washington, D.C. then, as well as some other meetings, the European counterpart as well. From there, moved on to CRISPR therapeutics, and then I feel like the rest is history. I've been sort of ingrained into the rare disease space, cell and gene therapy field. I've been fortunate to have amazing experiences at multiple companies now, either working on ex vivo cell therapies, in vivo gene editing approaches, in vivo gene therapy approaches using viral vector tools. I've been able to consult for companies using non-viral vector technologies. So I've really been sort of at the forefront of many different technologies.
Wow, that's quite the journey, right? From West Virginia to the pit, to New York, to Boston, and also the early days of gene editing. That's super cool. And, so going through all these stages in your career, what's been really the most rewarding about working in the field?
Oh, great question. I think, ultimately, it's been very rewarding to be associated with programs that made it to clinical testing and now some of the approved drugs like Casgevy. Ultimately, I think we're all here for patients. And so having very small contributions to some of those programs directly and being a part of a field that's working in earnest to bring forward more therapies for patients has been very gratifying. I think, that's balanced, I think, also with, early discovery, there's a lot of failures. And so, one thing that's always been motivating to me is working with great people. And I think this field attracts a lot of talented entrepreneurial innovators. Sanjay and I, of course, work together closely at CRISPR Therapeutics, continue to work together as volunteers here for the Issue podcast. So I love all the relationships I've built in the industry over the years, including academic collaborators, patient advocacy groups, etc. It takes a community to bring forward therapeutics, and I love being tapped into that community.
Oh yeah, that's fantastic. And as you've grown and as your career is taken off right over the past 10 years, I was wondering what lessons you've learned from leading teams, cross-functional teams and programs, from early discovery into the clinic.
Oh yeah, really good question. Many lessons learned. Learning from your mistakes is always key. I think I approach most problems. So much of what we do is team-based, and I think the thing that I find most effective is starting with respect and empathy towards my colleagues, and being really clear the goals that we're setting, what's expected of individuals and those contributions, and trying to align resources around the problems at hand to move things forward. And I think that empathetic view really helps you to see how you can tap into other talented individuals to really let them excel at what they do and contribute to the problems at hand. And I think a lot of that involves really effective communication. So I love picking up the phone, walking down the hall, traveling across the world if it's needed, so that I can meet with a given team or individual to figure out the solutions to our problems.
Oh, that's awesome.
All right, Sanjay. I think it's time for me to turn. I think we've heard enough about me. I like to think that I'm humble, although in this podcast, obviously, we're putting ourselves out there probably a little bit more than we're used to. What about you? What sparked your interest initially in science, Sanjay?
Thanks for asking the question. Yes. I think it really started with biology, right? Especially animal behavior and evolution. My dad was a zoologist. He was a zoology professor, and growing up around him naturally sparked my curiosity about animals and just how living things work. So I grew up in Bangalore in India, and had the opportunity to do a summer internship after the first year of my master's program in zoology. And this was a place called the National Center for Biological Sciences. It was in Bangalore. And it was just for a summer, and that was my first real exposure to molecular biology, right?
No, those summer programs are really important.
Yes, and life-defining, I feel, career-defining for me, because I ran my first agarose gel there, stained with ethidium bromide, and I was completely fascinated by genetics and DNA. And that experience hooked me really early, and ultimately set me on the path toward biotechnology and therapeutic development. So that's how I got my start. I was always fascinated by DNA, and it was really this trying to figure out how living things work and how cells behave that really got me into science. It was mainly the biological aspect of science that kind of got
Interesting,
me into it.
yeah.
Yes, yeah.
So your foundation's in DNA. So how did that lead to your current career in the cell and gene therapy space?
Yeah, I've always been fascinated by DNA, not just how genetic information is stored, but also how it gets damaged and how it gets repaired once it's damaged. And I found it fascinating that the cells have these elaborate mechanisms to repair damaged DNA. And DNA damage happens all the time, just as a consequence of living and breathing. So during graduate school and my postdoctoral work, I focused heavily on DNA repair mechanisms. I worked in Graham Walker's lab at MIT for a number of years. And I was looking for a way to kind of apply what I've learned into a technology that would be useful for patients, right? Then CRISPR emerged, and what immediately struck me was that the entire technology fundamentally relies on the cell's own DNA repair machinery, right? So it felt like an incredible convergence of everything that I had trained in scientifically. So when CRISPR Therapeutics was getting started, I jumped at the opportunity to help translate this groundbreaking technology, right, into medicines. The chance to move from understanding basic mechanisms of DNA repair to actually developing potential cures for patients was incredibly compelling. So that's how I got into CRISPR. It was an extremely exciting time because there were a couple of other CRISPR companies that had started up, and it was really intriguing for us to see how this technology would actually be translated into the clinic.
Yeah, during your time at CRISPR, you were able to contribute to multiple programs that are now in clinical testing.
Yeah,
that must have been an amazing experience. Any lessons learned during your time working on those drugs?
Yes, I was really fortunate. I consider myself extremely lucky and very fortunate to be part of these program teams at CRISPR. Seeing these programs right from the really early discovery stages, right, into the clinic. Seeing this journey from early discovery, right, and to administrate and finally to a sort of therapy that's actually administered to patients in clinical trials was both humbling as well as very meaningful for me. I think what stands out also is how collaborative drug development is. It's never a one-person thing. It really does take a village. It's cliched as it sounds, does take a village. Working alongside experts in CMC, in regulatory, IP, farm talks, clinical development, and manufacturing gave me, a real appreciation for how many disciplines have to come together to translate an idea into a medicine, right?
Absolutely.
So I consider myself really fortunate. These cross-functional experiences have probably taught me as much as the science itself. So I just consider myself very lucky to be part of these programs.
Yeah, very exciting. And obviously it's great to see your contributions now in clinical testing. We're at the ASGCT meeting, lots of innovative science out there being presented, a lot of it still in the preclinical space.
Yeah.
What aspects of the field really excite you the most today, both in the clinical side as well as at this innovative edge of the ASGCT meeting?
Yeah, I think it's these waves of innovation that come. Keeping in touch with the technology is something that's always been a huge challenge for me. And places like a conference like this, right? Gene editing is such a rapidly evolving field, and in a conference like this, we get to see every single new emerging technology that's happening and how we can build and learn from it, because science and gene therapy in general is always based on precedent, and learning from these entire group of innovators and entrepreneurs is really the most exciting part of the field right now.
Excellent. One thing we wanted to talk about today is career advice. Sanjay, what's the best career advice you've received during your time in industry?
I think for me it's been networking. When I spoke to people who, as I've been in the field for a while, long-term relationships I noticed, and building trust with people matter more than a collection of LinkedIn contacts. And some of the best opportunities come from genuine scientific curiosity and staying connected. And conferences like this, like the ASGCT, essentially help us network with various people. Ultimately, networking is based on trust, right? You trust people, you collaborate with them, and you build the scientific community and professional community. So I met several people in the DNA repair field several years ago, and I still keep in touch with them. They tend to be my mentors. I network with them very regularly. So I think the best career advice I've gotten was to never stop networking and building that sort of trust with your peers.
Interesting. You mentioned, I might be going a little out of turn here, but you mentioned mentorship, and I think obviously good mentorship can help develop one's career so much, but often it's a challenge to identify good mentors. Can you speak to your experience with mentors and mentorship, potentially also mentoring others more junior to you as well?
Yeah, that's a great question. I think mentoring is a really important part for anyone's career, right? I had this a fantastic mentor in graduate school who happened to be the chair of my committee, and what he taught me very earlier on was the importance of focusing on the science, right? When things around you are not ideal, or if you're going through a hard time. And to not to worry about all the noise that's around, and to focus on what challenges lie before you. It's something that I learned a lot from, and I'm very grateful for that sort of mentoring opportunity, because he not only taught me how to do science the right way, but also how to deal with difficult people, with collaborations, and placing trust in people you may or may not know, right? So I think mentoring is a really important part of anybody's career.
That's really interesting.
I'd like to turn it over to you in terms of mentoring. I mean, how do you feel about mentoring and?
Yeah, you brought up tough times, and I think whenever you're working under tight timelines, trying to move either to the next milestone, or trying to get regulatory filings at a set date, when you come under pressure, there's so many moving parts. So many different people involved, different egos, different backgrounds. It's always a real challenge, for even the best working teams to do those things seamlessly. And one of my early direct managers in biotech at Amgen, Cindy Wilson, was also a great mentor to me as I transitioned from academic science into industry-based science. And she gave me advice that I still think about so often today, and that was always assume the best in people. Everybody has different things going on in their personal lives, professional lives. Everyone's at a company is working towards these common goals. So, whenever you're in those tough times, just really clearly articulate goals, clearly, say what you're going to do, try to motivate, help others that are working with you towards those goals. And, if the motivations of other people, are things that you can speculate on all you want, but I think it's just such a healthier life and a more rewarding experience in general if you're just always assuming the best in people, trying to empower people to do their best, also be an example of that by doing your best. On the subject of mentorship, it's tough. I think particularly at smaller biotechs, less resources. We talked about this offline to some extent. I think mentorship often takes the form more of this apprenticeship where you get to witness a lot of decision making, wear a lot of hats at a small company, but managers and leadership don't often have the time to take these one-on-one personal relationships where they get to take chunks of time to dedicated mentorship where it's a two-way conversation about what's working, not working. So often in those small biotechs, people have to look outside their organization maybe to find meaningful mentorship, and that can be challenging. You have confidentiality rules, etc. So maybe those relationships are somewhat constrained. But that's where that networking that you brought up is so important, like trying to find individuals both either within your company or outside your company that can serve a mentorship role where you can have those meaningful conversations that has both the good, the bad, and the ugly, right? Being open to all of that ultimately establishes the most powerful mentorship relationships in my experience, either as a mentee or as a mentor, where again, as long as it's based on this foundation of respect and empathy, I think, being able to exchange ideas around what's working, what's not working, being honest with yourself about what motivates you. Those are the types of conversations that can really help build one's career in a way that's positive for yourself as well as positive for your colleagues and those around you. I found, again, in general, for me, my personal experience is it was a lot easier. The larger companies I had, I felt like you had so many different peers different layers of management, it created the space to find probably more people to tap into that also had, all teams are under tight deadlines, but I'd say in general, at larger companies, they do, carve out more time for that type of development and mentorship. So I felt like I was very lucky to have that foundation at Amgen, which was my first industry job, and I've tried to bring that with me as I've moved on to other opportunities at companies, either working under extremely tight resources, extremely tight timelines, under-resourced teams, trying to wear many different hats, research, development, regulatory IP, CMC, what have you. But it's something you have to work at continuously. It's not like I found a mentor 10 years ago and, just had that relationship continue. It's something I've had to build, reboot as needs arrived and different levels of mentorship are needed. So it's something I continue to work on. And I've been lucky also to, try to mentor others that I've worked with through the years. And again, sometimes I feel bad because the schedule gets tight, meetings and lunches dedicated to mentorship fall off the calendar. And I think we all recognize the importance, but figuring out how to prioritize it is always an issue.
Let's focus on the issue podcast, taking over from Emily. What excites you about it? And
Yes, Sanjay. Obviously, really excited to be taking over the issue. Little daunted. I think Sanjay and I both don't have podcasting in our blood and veins. Emily reached out thinking that we could be the next host based on our time working together over the last 10 plus years, and so I'm really looking forward to working with ASGCT and using this as a forum to really dive into the issues in cell and gene therapy development. Specifically, I'm really looking forward to curating some shows around AI. I think everybody's thinking about AI and how it can be incorporated into their work streams, whether they're at the bench, whether they're thinking about regulatory filings, clinical trial design, what patients their drug could be used for. I see and hear examples of AI being used across that. But myself, I have not adopted AI into any of my work streams, so I don't want to be on the outside looking in either. So maybe we can use this as a forum to really explore that through several episodes, looking at it from different perspectives with guests who know a lot more about it than us. I'm also really looking forward to all of these cell and gene therapy initiatives out there for ultra-rare diseases, particularly those initiatives maybe falling outside of the traditional biotech and pharma model. We'll be talking to Fyodor Ornov, who led one of the really exciting applications of base editing to an ultra-rare patient population. I think those types of conversations are really interesting. So much to learn, so much still to do ahead for the field in those areas. Currently, I'm employed at a China-based company that's public on the Hong Kong Stock Exchange, Cambridge Pharmaceuticals. So I'd love to highlight some of the innovation going on in Greater China, both preclinically in the lab at both academic and biotech companies, as well as clinically, with investigator-initiated trials for the cell and gene therapy space, creating such high value for what potential therapies can do in some of these rare and potentially ultra-rare patient populations. And it's a tool I think a lot of companies are using to try to de-risk their medicines and also have impact on patients in China who may not have other opportunities for therapies otherwise. I'm also fascinated by some of the cutting-edge applications for cell and gene therapies, particularly as cell and gene therapies are de-risked more and more clinically. I think applications towards higher quality of life or longevity. There was recently some New York Times articles around this idea of longevity research and some of these programs going to the clinic. Obviously, there's a underbelly to that world, these sort of snake oil salesmen that are sort of over-promising what those therapies can do. But I think there's a lot of legitimate, high-quality, rigorous science going on in this field at places like Altos Labs, and maybe their name, I'm not sure what their exact name is, in the Bay Area. Companies like Life Biosciences that has an IND open on an eye application for using reprogramming factors to hopefully repair optic nerves. I think there's a lot of exciting work at this very cutting edge of the field, both in the clinical and preclinical space. Would love to bring some of those topics to the podcast as well. What about you, Sanjay? What are you looking forward to doing here at the issue with ASGCT and the great team there?
Yeah, I too am really excited, and I'm really excited to cover the topics that you brought up, Jason. I think one theme across all shows will be definitely trying to do our best to use the podcast to bring together people in academia, in biotech, clinicians, regulators, patients, patient advocates, as well as commercial considerations, right? And I'd like to create a space for all of these people, both in terms of not only the drug, but people advocating for the patients, the people using the drug, and also a space for established mentors and rising voices, right? Particularly those that we haven't really heard of, or the lesser known ones. Long-term patient data follow-up, as scientists and drug makers in biotech, it is really our duty to educate the world on our successes as well as failures, because great science and breakthrough medicines are always based on precedent. I was struck by what Kiran just spoke about, about being your own worst critic as far as drug development goes. We'd like to see successes. We also like to see failures so that we could learn from the successes as well as failures. Right?
That's great. I think you're going to do a great job filling the shoes of Emily, because I think Emily set a great framework for that in the podcast through the last 3+ years, because I think she did such a great job meaningfully covering all those areas. And obviously that's the big shoes to fill there, because covering all those areas meaningfully is a challenge, and it takes a village, right? Like all things. And so that's why we're co-hosting. Sanjay and I can hopefully complement each other's strengths and weaknesses together. But we also want to tap into the expertise, obviously, of the ASGCT, but as well as our listeners. So we wanted to use this as an opportunity to ask our listeners, what do you want to see covered in the issue? We'd love to hear from you. Topics, people, different perspectives, either perspectives professionally or personal perspectives and backgrounds. We want to try to embrace all of that diversity here in the podcast. You can reach out to us via email at media@asgct.org, or you can find Sanjay and I on LinkedIn. It is useful for networking to a certain extent, Sanjay.
Yes,
it's also really useful for communication. You can get in contact with ourselves there. And ASGCT also posts actively on LinkedIn, so please follow. I suggest everyone follow ASGCT there, so you can learn about when new issue podcasts come out, as well as some of the other amazing podcasts that ASGCT is curating and publishing. Any closing words, Sanjay, from here in Boston at ASGCT 2026?
I certainly, if you're interested in joining us as a guest, please do reach out. I think some of the best conversations I've ever had come from very unexpected perspectives. So I'd love to hear from you. If you're interested in joining as a guest or even co-hosting a future episode, please let us know.
Awesome. Great talking to you, Sanjay.
Likewise.
Look forward to doing many issue podcasts with you, as long as ASGCT continues to let us. We're volunteering, so it's hard to fire us, probably.
Yes.
So hopefully you'll continue to tune in and listen to us. And hopefully it'll be more interesting as we bring in more guests other than Sanjay.
Yes. Absolutely.
Thank you.